All right, welcome everyone. In this week's lecture, we're going to talk about the effector mechanisms of humoral immunity, and this will close out our multi-chapter series on the adaptive immune system's activations and mechanisms. And then afterwards, we're going to get into tolerance, autoimmunity, transplantation, and cancer immunology. So let's jump into it. Now, I think to help understand the functions of the humoral arm, it helps to understand the differences and purposes between cell-mediated and humoral arm first. So let me first give a quick review on cell-mediated immunity. So the adaptive cell-mediated response is mediated by T lymphocytes, and these protect against intracellular pathogens and tumor cells. We learned that there's two main branches of T lymphocytes. Those are the helper CD4 plus cells and the killer CD8 plus cells. So the CD4 plus cells play a critical role in all phagocytic elimination of microbes. Unlike CD8 plus cells, CD4 plus cells use cytokines to primarily mediate their functions, and they have an essential role in activating other leukocytes like eosinophils and neutrophils, and as we learned in the last chapter, in antibody production and activation of B cells and producing higher affinity and long-lasting memory B cells. CD8 plus cells, on the other hand, don't necessarily help other cells, but they directly kill infected cells. They do this through direct cell-cell interactions, as we learn the release of perforin and granzymes through the immune synapse. However, they can also indirectly mediate their action by the secretion of IFN gamma, which induces phagocytes to kill their endocytosis microbes. Now, as we get into humoral immunity, we can highlight some of the differences between those two and then get into detail exactly what B cells and their antibodies, which mediate their functions, actually do. So again, humoral immunity is now mediated not by B cells, but actually by the secreted antibodies. So think of the B cells or the plasma cells as factories and the antibodies actually carrying out their functions. Okay, and these are responsible for the defense, the body's adaptive immune defenses against extracellular microbes and their toxins. Now, unlike T cells, which have to first be produced and then migrate to the site of the infection, the inflamed piece of tissue, and perform out their effector functions, B cells and plasma cells don't have to do this. Right? So the antibodies are the ones that perform their effector functions at sites distant from their production. So usually we want to think of antibodies as being produced in the bone marrow. That's where the long-lived plasma cells that are developed through T-dependent mechanisms, activations, also inflamed tissues, secondary lymphoid organs where antibodies can be produced. There's also the concept of tertiary lymphoid organs. I put that in quotes because it's not an actual organ or part of the lymphatic system in the body, but at sites of chronic inflammation, the body will actually develop germinal centers that develop these factories almost on site, and it's usually a signal of like a runaway immune reaction, like in cases of chronic arthritis is a classic example. So it's a bad thing. And it's not actually part of the lymphoid organ. So again, that's just why it's in quotes. One cool thing that differentiates humoral immunity and antibodies from T lymphocytes is that their mechanisms can access locations that are otherwise inaccessible to other parts of the adaptive immune system. Okay, so things like transepithelial locations like the GI tract, the respiratory tract, transplacental, right? So we already learned that IgG antibodies can attach to the Fc neonatal receptor and get into newborns in ways that T lymphocytes or other cells aren't able to. Now, as we get into the effector functions of antibodies, two things I want to point out as far as the antibody structure. So we learned previously that the selectivity of antibodies is provided by the fragment antigen binding area, FAB. So that's the area that undergoes somatic hypermutation, gets the affinity maturation, and the part that actually binds to the antigen. However, the effector functions of antibodies are actually mediated by the common domain, the FC portion of the antibody. So this is the part that eventually gets everything done. Okay, not the antigen binding part, just that little stem part of the antibodies. We already kind of knew some of this. We knew that Ig1 and Ig3 antibodies bind to the phagocyte Fc receptor to promote phagocytosis. So that's the Fc portion that binds to the aptly named Fc receptors. We have some understanding of IgM, Ig1, Ig3, to a lesser extent IgG2, of activating complement, and we're going to talk about complement a lot more in this chapter. And then IgE binding to mast cells in eosinophils. So they will have actually already bound through their antigen binding portion to the helminths or other parasites that they're meant to attach to. And then it's the Fc portion that actually activates mast cells or eosinophils to degranulate. Now, a very good question at this point would ask, okay, if it's the Fc portion and we have these antibodies floating around in our circulation, why aren't these Fc portions just binding to receptors randomly and just triggering degranulation? And it's an excellent question, because the follow-up is that the effector functions require the binding of an antigen to the variable region. Okay, so these Fcs actually can attach to the receptors unless they're oriented in a very specific way, and they cluster in a specific way that wouldn't happen unless they were actually bound and in that more organized pattern. Okay, so it brings these antibodies in clusters together, repeating epitopes that allow the bonding of multiple Fc receptors on a particular cell. And this also helps ensure that the effector functions are only carried out when needed. So you can have antibodies floating around that are just benign, and they have their Fc portions that could potentially bind to receptors, but it just doesn't happen in the right way, in the right concentrations, in the clusters that's necessary, because there's no antigens there to bind to. Now, there is one significant exception to this rule, in that the Fc portion carries out the effector functions of antibodies. And that's in the case when antibodies neutralize toxins. Now, neutralization is simply the binding of the antibodies to a particular toxin or even pathogen that just simply blocks them from doing whatever they want. Maybe it attaches to a receptor, maybe so many antibodies attach to a toxin that it can't then perform out its functions just due to steric hindrance. So in that case, the Fc portion is really just getting in the way, but not actually doing anything itself. So neutralization and just the binding itself of antibodies can itself produce an effector effect without the Fc portion doing anything notable. So now let's talk about some of the antibody functions in detail, because there's a number of different roles that antibodies can play within the body. So I just talked about the idea of neutralization, and it's really the simplest of all the function. It's where the antibody simply blocks pathogens or the toxins from acting just by coating the outside of it. So this part only requires the Fab region of the antibody in order to carry out their functions. And despite being so simple, it's incredibly effective, and we use this a lot for a lot of different vaccines and different treatment options. So some notable examples are for the polio virus, which forces our body to produce both IgG and IgA antibodies. There's the tetanus and diphtheria toxins, in which IgG just attaches to them and then prevents them from acting on toxic effect in our body. In our fight against hepatitis A and hepatitis B, our body primarily fights it off through the use of antibodies that just neutralize the virus. Yes, we do produce CD8 plus cells that have a big damaging effect to our body, but it's primarily the antibodies that fight off and stave off the infection. And also flu viruses. Whenever you get a flu vaccine, we primarily intend to neutralize the virus as opposed to, again, producing CD8 plus cells or CD4 plus cells to help out with other functions. We've talked about the concept of opsonization, certainly in innate immune reactions and what complement carries out. Antibodies are fantastic opsonizers. So in this case, opsonization is a fancy word for just coating a particular microbe. And then phagocytes can recognize the opsonins like antibodies, and antibodies are really powerful activators of phagocytosis. We learned about the Fc gamma receptors. We're going to talk about those in more detail today. So in addition to inducing the intracellular killing mechanisms through phagocytosis, they can also cause degranulation and other mechanisms that are induced by other cells to take an effect. So here's a little video from the book. I think that goes into this topic a little bit more detail. So let's take a look at that and then get back to the rest of the effector mechanisms.
Antibodies coat microbes and promote their ingestion by phagocytes. The process of coating particles for subsequent phagocytosis is called opsonization, and the molecules that coat microbes and enhance their phagocytosis are called opsonins. When several antibody molecules bind to a microbe, an array of Fc regions is formed projecting away from the microbial surface. If the antibodies belonging to a certain isotype, for example IgG1 and IgG3 in humans, their Fc regions bind to a high affinity receptor for the Fc regions of gamma-heavy chains, called Fc gamma Ri. Signals from the Fc receptors promote phagocytosis of the opsonized microbes. The phagocyte extends its plasma membrane around the attached microbe and ingests the microbe into a vesicle called a phagosome and activates the phagocyte to destroy the microbe.
Now that we've talked about some of the opsonization mechanisms, let's go into a little bit more detail on the actual receptors involved in this process. Now, the first one I know we've heard before is the Fc gamma R1 receptor. I want us to think about this as the major Fc gamma receptor out there. It's going to be the one with the most potency. It has the highest affinity towards the Fc portion of an antibodies. It binds specifically to Ig1 and Ig3 to a greater extent than other antibodies. It's the most potent phagocytosis initiator, and it's found primarily on macrophages and neutrophils. There are three different kinds of Fc gamma R2 receptors, named A, B, and C. All of them have very similar extracellular regions. They bind to the same ligands or very similar ligands, and they really just differ on the intracellular components and what they actually signal on the inside of cells. The A and C receptor, in general, are both activators of phagocytosis to a lesser extent than the Fc gamma R1. They're expressed on neutrophils, any mononuclear phagocytes, so monocytes and macrophages, dendritic cells to the A receptor, a little bit more so than the C receptor, and vice versa for natural killer cells. Just like Fc gamma R1, they tend to bind to IgG1 and 3, not 2. And they have a lower affinity, about 2 to 3 orders of magnitude less than that of the Fc gamma R1. So that kind of gives you an indication of the relative roles of each of these receptors in phagocytosis. The Fc gamma R3 receptors, which come in two different forms, A and B, have very similar extracellular domains as the Fc gamma R2 receptors. But again, they have different functions and they're found primarily on different cells. I want us to associate both of these mostly with natural killer cells, particularly the A. The B receptor is primarily associated with neutrophils, although its mechanism is poorly understood. The A receptor is primarily responsible in the antibody-dependent cell-mediated cytotoxicity functions of natural killer cells. They do this by primarily binding to the IgG subclasses of antibodies. The Fc gamma R2 B subvariety of this receptor we learned about previously in its role in antibody feedback. It's the only receptor, the only Fc receptor, that B cells express. Oddly enough, other non-lymphocytes tend to express this as well for other functions that we're not going to get into. But it's the only Fc receptor that B cells responds to, and it's used in antibody feedback. So whenever there's a high amount of this particular receptor on B cells, then the ability for these cells to bind to the Fc antibodies inhibits further production and activation of their antibodies. So the big difference here is actually just the intracellular domain. So the extracellular are exactly the same. In fact, they even have the same cluster differentiation nomenclature. They're both CD32. And it's entirely their intracellular domains that are different. They lack the ITAM, that sort of red portion that the Fc gamma R2 A and C subvarieties have, and instead have an inhibitory mechanism on the inside of the cells. And then finally, we're going to come back to the Fc neonatal receptor, which we talked about, I think, way back in chapter 5. This is expressed on a variety of cells, expressed all across the epithelium, the placenta, a few macrophages, and other type of phagocytes. So we want to associate this with not just transport across the epithelia, but also with phagocytes, because that's how our body naturally processes some antibodies to get rid of them. IgG antibodies can bind to this and get protected from the process of lysosomes and phagocytosis by escaping that route through this particular receptor. That's why it's a pH-dependent route. Endosomes drop in pH, they acidify, and that's what increases the binding of these antibodies to that particular receptor, and then protects them from degradation that way. And that's why they have such an increased half-life in comparison to the other subclasses of antibodies. There's two other receptors that I want to quickly mention that I don't think the book really touches on in this particular subsection, but I feel make the honorable mentions list here. The first is a polymeric Ig receptor, aptly named PIGR, that in general binds to a few different types of antibodies, the Fc portion, and transports them across epithelial layers. So it's really important in gut immunology and immune functions at mucosal areas. So I want us primarily thinking of IgA and IgM functions there. That's the antibodies primarily responsible for our immune defenses in those particular areas of the body. And that's how they actually get across. So plasma cells will produce them in the bone marrow, and they end up at the epithelial mucosal areas because of this particular receptor. Then there's also the high affinity IgE receptor, yet aptly named Fc epsilon Re, so epsilon E, that's how I remember it. This is the one that's expressed on mast cells, basophils constitutively, and also it can be induced in eosinophils, and they're the ones that release all of their granules to kill helminths, for instance. And that's why they bind to IgE, and that's why we associate IgE with the eosinophils and mast cells to a lesser extent as well. Now, we had talked about phagocytosis in general in Chapter 4 in the innate immune functions. So I want to tie it back here now that we're talking about antibodies and tie it to the Fc gamma receptors. So the general order of process things here is that first the antibodies would opsonize the microbe, primarily by IgG. That's what we want to associate the Fc gamma receptors with. Then we have the binding of the opsonized microbe to the phagocyte Fc receptors. In this case, Fc gamma R1 is the one we want to think of as the most potent one. That Fc signals a number of different things, including the activation of the phagocyte and the endocytosis process, so engulfment of the opsonized microbe. So then we have the phagocytosis process and the killing of the ingested microbe by the killing actions, reactive oxygen species, hydrolytic enzymes, nitric oxide, and so on. Within this category, again, I want to reiterate that Fc gamma R1 is the most important receptor for phagocytosis. And then the IgG1 and IgG3 varieties of antibodies are the most efficient opsonins and activators of phagocytosis. And just like how we talked about the activation of cells in general doesn't just require one antigen, one receptor, but a cluster and many of these receptors bonding at once, activation of these phagocytes requires multiple cross-linking of Fc receptors by several adjacent Ig molecules. That's one of the ways, again, that the Fc functions are carried out only when bound to antigens. So antigens might have a number of different antibodies bound to them, all in the repeating epitopes that then allow for the clustering of these antibodies. So it's the exact same way that the lymphocytes themselves get activated, phagocytes need to be activated by multiple binding of these receptors. The binding in any of these receptors in general can produce a number of different changes. So first of all, there's the secretion of cytokines that help activate neighboring and adjacent cells. There's the release of inflammatory mediators and microbial enzyme secretions that act on the extracellular space. There's the general mobilization of the cell wall, in which we think of engulfment and endo and phagocytosis. And then there's also the release of granules. So again, thinking of mast cells and eosinophils, the release of their granzymes to the extracellular space. A lot of the killing actions within inside the cells are also initiated by the binding of these receptors as part of phagocytosis. So again, the production of reactive oxygen species, nitric oxygen species, and hydrolytic enzymes. So that covers the opsonization and phagocytosis part of antibody functions. Now we get into antibody-dependent cellotoxicity. So this is what we associate with natural killer cells. We want to associate this with the Fc gamma R3A receptor. And the result of the clustering of antibodies with this particular receptors is the release of the granules like perforin and granzymes from natural killer cells that mediate the cell-mediated cell death, and then also the release of IFN gamma to a lesser extent for the cytokine-mediated cell toxicity. So now I want to talk about a series of experiments run by the Belgian scientist Jules Bordet in the late 1800s. So in general, scientists knew of the ability of serum to have these, what they thought were antitoxins at the time, which we now know are antibodies, to potentially treat people with infections. So in this particular experiment, they had this petri dish of a certain bacteria. They knew they had serum that contained these antitoxins specific to these particular bacteria. And then if they treated these bacteria with that anti-serum at 37 degrees, then that result will be that the bacteria dies. Now, as a follow-up experiment, Jules Bordet repeated the same thing, except this time he heated the serum up to 56 degrees and then brought it back down to 37 for the treatment in the petri dish. Now, what was interesting about this is that they knew that whenever they could isolate these antitoxins, again, these antibodies are actually very stable compounds and that they're going to survive relatively high temperatures. Yet in this particular experiment, when they did this and they heated the serum up and then treated the bacteria with it, the bacteria, it failed to impact it at all, and the bacteria lived. They were just fine. So again, this baffled scientists in that they knew that antibodies should be able to withstand this, yet it didn't cause the same impact at the as the experiments at the lower temperatures. So Jules Burdett repeated the same experiment, except this time, instead of using petri dishes with the bacteria, gave the bacteria to an infected mouse, or infected a mouse with that bacteria, did the same thing with the serum samples from an already immunized mouse. In this case, they had serum with the antibacterial antibodies, heated that serum up before delivering it to the mouse, and the net result is that the mouse got treated. So to recap, they knew they had antibodies specific to a particular bacteria, so they knew that it would kill bacteria in a petri dish. They also knew that whenever they heated the serum, it somehow wouldn't work, even though that they knew that the antibodies were stable at that temperature. They also knew now that if they heated that same antibody solution and gave it into a live specimen, something that already had serum, that it would still somehow work again. So the main conclusion from this is that serum, so in this case the infected mouse would already have, has some sort of heat-liable components that complement and help the antimicrobial functions of antibodies, which is where we get the name complement from. And for these particular sets of experiments, Jules Burdett won the 1919 Nobel Prize in Medicine. So in general, complement are just a series of plasma proteins that help mediate effector functions for both the innate and adaptive immune system. Now, we always have these complement proteins in our system, yet they don't do anything because they're only activated by microbes and antibodies. The activation of complement results in these proteins turning into proteolytic enzymes called zymogens that continue this cascading process that amplifies further complement activation. So a single zymogen can convert multiple other proteins, in this way act as sort of this amplifying effect. Once cleaved, these proteins covalently attach themselves to microbes and antibodies, and it's only in this form that they're actually activated. What's nice about this is that in the fluid phase, complement proteins are really inactive or only transiently active for a few seconds, and it's only once they actually bind to something that they're stable. This helps ensure that their effector functions are only carried out once they've bound to antibodies or microbial surfaces, and not just in the free plasma. Whenever complement proteins are cleaved, some components of it turn into the proteolytic enzymes, the zymogens. Other components turn into byproducts that activate and stimulate inflammatory reactions. Now, as the initiation and cascading effect of the complement activation process requires some sort of surface in order to happen, host cells have developed regulatory mechanisms that inhibit complement activation. It's the fact that microbial and biomaterial surfaces lack these regulatory proteins that continues complement activation, whereas host cells wouldn't do it. Now, we've talked about complement in previous chapters, so I want to give another brief overview of the three different complement pathways. So there are three major pathways in which complement can be formed. So the classical pathway actually evolutionarily came later than the alternative pathway. Classical pathway is antibody mediated, so that's what we talked about with Jules Burdett's discovery. The alternative pathway is actually what we as organisms developed first, and that's an antibody independent pathway. And then there's also a lectin pathway, which is very similar to the classical pathway. Instead of using antibodies, it uses a mannose binding protein called lectin. Now, the important thing I want to highlight first of all is that all three of these processes result in the cleavage of the complement protein C3 into its two subcomponents, C3a and C3b, by the enzyme C3 convertase. C3b is what becomes covalently attached to the microbe and antibody surface, and this is what gets recognized by phagocyte receptors. C3b is also involved in the conversion of C5 complement protein into its two subcomponents, C5a and C5b, by C5 convertase, and then so on continues the complement pathway. I'm again going to let the book continue on with a video that kind of discusses some of these pathways and the process towards complement completion, and then we'll talk about some of those steps a little bit more in detail.
The activation of the complement system may be initiated by three distinct pathways, all of which lead to the production of C3b, the early steps. The alternative pathway is triggered when some complement proteins are activated on microbial surfaces and cannot be controlled because complement regulatory proteins are not present on microbes. The classical pathway is most often triggered after antibodies bind to microbes or other antigens and is thus a component of the humoral arm of adaptive immunity. The lectin pathway is activated when a carbohydrate-binding plasma protein, mannose-binding lectin, binds to terminal mannose residues on the surface glycoproteins of microbes. Activated complement proteins function as proteolytic enzymes to cleave other complement proteins in an enzymatic cascade that can be rapidly amplified. The central component of complement is a plasma protein called C3, which is cleaved by enzymes generated in the early steps. The major proteolytic fragment of C3, called C3b, becomes covalently attached to microbes and is able to activate downstream complement proteins on the microbial surface. C3b initiates the late steps of complement activation, culminating in the formation of a multi-protein complex called the membrane attack complex, which includes C5b, C6, C7, C8, and C9, and causes lysis of thin-walled microbes.
So I thought that was a great overview video of the entire process from start to finish. I want to highlight some of the main differences between the three pathways, so particularly how they start, and then talk about some of the functions at the end of what complement actually does. So first, let's start with the alternate pathway. It's the oldest and simplest complement pathway. And the way it works, because there's no initiating factor like with the mannose binding lectin or with antibodies, is that C3b is naturally and constantly cleaved and generated at a very slow rate, even in healthy individuals with no foreign objects or antibodies or mannose binding lectin within them. It's a process called C3 tickover. Now, as I mentioned previously, C3b can only covalently bind to a surface if it's foreign and if there's no inhibiting proteins. So if there are microbial surfaces around, C3b is naturally cleaved in just the free plasma, so it's free to then bind to microbial surfaces or foreign surfaces. Otherwise, like I said, if it doesn't bind to anything, it becomes inactive because it hydrolyzes very readily in just free plasma. It's very unstable unless it's bound to something. So always remember that even healthy individuals are always generating C3b, just nothing happens because it doesn't have anything to bind to. The classical pathway instead uses antibodies to initiate a few precursor steps that actively generate C3b in a way that the alternative pathway skips right to. Okay, so think of the classical pathway as having a few added steps that occur before the formation of C3b that are required for the formation of C3b. And it's all initiated by the very first complement protein complex called C1. The C1 complex is a combination of a few different proteins that are combined together. So the C1q is the largest of them, which is this hexamer protein that within its sort of arm folds has two each of the C1r and C1s proteases. And it's the C1q portion that binds to either IgG at its second heavy chain or IgM at its third heavy chain portion of the Fc part of the antibody. And again, I'm going to stress that this can happen to free-floating antibodies. These antibodies have to be bound to the antigen so that their Fc portion sticks straight up in this conformation that allows for the C1q binding. They can't just freely float around and just happen to all six simultaneously bind. It's just, statistically, it just doesn't happen. Upon this binding with the antibody in the right conformational state, that initiates the activation of both the R and the S subtype proteases that cleave the complement proteins C2 and C4. Next comes the lectin pathway, which I like to think of as the antibody pathway or the classical pathway, but in disguise because there are a lot of similarities. So I just want to cover exactly what's different about them and what's similar about it. The body has a number of different circulating lectins that bind to carbohydrates and polysaccharides that are typically unique to bacteria. A common one that we've touched on before is the mannose-binding lectin, MBL, and this sort of structure after it's bound to its polysaccharide is very similar to C1q. Now, just like C1q binds to the C1s and C1r proteases, mannose-binding lectin binds to MBL-associated serine proteases that act in the very similar way. In fact, what they do is the exact same thing as the classical pathway in that they help to cleave C2 and C4 complement proteins. Now, one of the main takeaways that I want you to come away with is that all these three pathways converge to the point where they develop a C3 convertase. Now, C3 convertase is not a unique molecule. In fact, it can take a variety of different forms. We see with the alternative pathway, since there's a natural C3 tickover that can actually, on a very low level, cleave C3 into C3b. The C3 convertase here actually has C3b in it, whereas the ones from the classical pathway and the lectin pathway, which are identical in this product, the C3 convertase is just a combination of C4b and 2a, some of the two byproducts of C4 and C2 cleavage. Now, at this point, all the three different pathways have a type of C3 convertase, which then cleaves free C3 into its byproducts C3a and C3b. C3b then will combine with the C3 convertase to form C5 convertase, which again takes a variety of different forms depending on how it was formed, either in the alternative pathway or the same byproducts from the classical and lectin pathway. Now, just as the purpose of C3 convertase is to cleave C3, the purpose of C5 convertase is to cleave C5 into its byproducts C5a and C5b. Now, in the later stage of a complement activation, C6 and C7 enter, and they begin to actually infiltrate the actual cell membrane instead of just covalently binding to the outside. C8 enters, and C8, as you can see in the pictures, actually goes through the entire membrane and starts forming these unstable pores around 0.4 to 3 nanometers, which is actually relatively large for a cell size. So this in itself, at large concentrations, is capable of lysing cells at high concentrations. C9 is itself a self-polymerizing protein that can build up these huge pores up to 20 nanometers in diameter. This is huge on the relative scale of a pathogen or cell. All of these proteins from C5 to poly C9 form the cytosol membrane attack complex, or MAC. So this creates this huge pore within the membrane that results in influx in water, osmotic swelling of the cell, and ultimately rupture and lysis of the cell. And in fact, C9 is structurally similar to the perforin molecule that natural killer cells and cytotoxic T lymphocytes, the CD8 plus cells, use to initially rupture and disrupt the cell membrane. Now, the previous slides all illustrate the entire complement pathway if it were left to just complete out as is. And many times that's what happens. That in itself can rupture the cell walls of bacteria and viruses and kill them in itself. But there are also other functions of complement proteins that actually take part in many of the mechanisms that we've discussed before, but we've just kind of ignored up until this point because they really just act as mediators, and we didn't have to get into it until now. So there are many receptors that activate on complement itself, and then will also work in conjunction with some of the Fc gamma receptors that we talked about. The first receptor we're going to talk about is complement receptor number 1, SR1. And the function of this receptor is to promote phagocytosis of bound obstinins that are bound with C3b and C4b. Oftentimes, the microbicidal mechanisms of phagocytes are activated whenever both these complement receptors are bound and activated, along with the Fc gamma receptors simultaneously. This receptor is also involved in clearing these immune complexes from circulation after they're no longer needed. Complement receptor number 2 has a lot of important functions associated with B cells. So in addition to B cells, some epithelial cells and follicular dendritic cells, which are sort of attached to B cells as well. They function to enhance B cell activation with certain antigens, and they also promote the trapping of antigen antibody complexes within the germinal centers that are then necessary for the somatic hypermutation and the expansion of long-lived plasma cells and memory cells during T-dependent activation of B cells. The type 3 complement receptor is also called MAC1, and we learned about MAC1 during the migration of leukocyte subchapter because it's also an integrin that binds to intracellular adhesion molecule 1 on endothelial cells. It's expressed on neutrophils, mononuclear phagocytes, mast cells, and natural killer cells. For neutrophils and monocytes, it helps promote phagocytosis of microbes that are opsonized with certain byproducts of C3b. There's a few other complement receptors that I'm going to quickly go over. The type 4 complement receptor, CR4, is very structurally similar to MAC1 and performs similar functions. The complement receptor of the immunoglobulin family, CRIG, is expressed on liver-specific tissue-resident macrophages called Kupffer cells that bind to complement proteins C3b and the inactivated form of C3b to help clear opsonized bacteria and bloodborne pathogens, specifically in the liver. And there's also receptors for the different complement fragments like C3a, C4a, and C5a, and these all help to, in general, stimulate inflammation. So that in general describes the complete pathway of this cascading effect of complement activation, the formation of the byproducts, and what cells have what receptors that can recognize any of these complement proteins. So I would say that the three major functions of complement are to promote phagocytosis of microbes, in which complement is activated, to stimulate inflammation, and then to induce the lysis of these microbes. Opsonization is primarily carried out by the C3b, C4b, and the inactive IC3b fragments, which primarily activate the following phagocyte receptors. So CR1, MAC1 or CR3, and then CR4, which is very similar to CR3. So CR1 recognizes C3b and C4b, and then both CR3 and CR4 recognize the IC3b. Now, CR1 is by itself a poor initiator of phagocytosis. It can happen to a limited extent, but it's augmented by the simultaneous binding of Fc gamma receptors. So we can sort of think of it as a weak reaction in the innate immunity, but then is augmented by the antibodies present in adaptive immunity. The cytokine IFN gamma also enhances the function of CR1-mediated phagocytosis. So up until now, we've talked about the phagocytic mechanisms in both the innate and adaptive immune functions, how B cells can help initiate that with antibodies, how T helper cells can initiate actions with phagocytes, and it's always been the intermediary that we've ignored, the complement protein C3b and IC3b that are involved in this particular defense mechanism. So now I want you to go back and think, every time I've learned about phagocytosis, we've been ignoring this, but now we know that it's been involved this entire time in all those different defense mechanisms. Now, the inflammatory byproducts of the complement pathway are all those little parts that don't actually take part in the formation of C5, C6, up to C9 and so on. So C5a, C4a, and C3a that we've, in all these pictures so far, seen that just kind of float off into the extracellular space. They're powerful inflammatory mediators that work by activating mast cells, neutrophils, and endothelial cells to, in general, induce small to potentially large amounts of degranulation by themselves. And all these degranulations act to cause vasoactivation, vasodilation, and the release of inflammatory products that mediate those, like histamine. Now, these products can either work by themselves or be incredibly augmented and potent when combined with mediators such as antibodies like IgE that act on the Fc epsilon R1 receptor, which I think we'll cover in chapter 20. And because these complement proteins are activators of inflammation, these fragments are named anaphylatoxins. The last major function, as I alluded to earlier, if you just let the complement pathway continue uninhibited without opsonization or worry about the inflammatory byproducts, then the formation of that membrane attack complex, MAC, on the surface of the pathogen is going to actually result in the lysing of those cells. Now, this is really only effective against pathogen with cell walls, which unfortunately is only a few amount of pathogens. So that's why our bodies evolved much more effective and more complex mechanisms that would take over in places where just the cell lysing mechanism would be insufficient. Other functions of complements serve to solubilize immune complexes that help clear them out. So we previously learned in the antibodies chapter that because antibodies have two bonding sites, it's possible at just the right concentrations that one antibody can link up two different antigens, and then another antibody can come on and link that antigen with another antigen and form these really large complexes. Well, the complement proteins serve to sterically block multiple and sort of linkage of multiple antibody antigen complexes, these immune complexes that would otherwise precipitate, form on the outside of cell walls, and continuously activate other complement proteins or potentially damage cell walls. So this helps keeps these immune complexes from forming and solubilized and allows the body to clear them more readily. Also, in the previous chapter, we learned about the activation of B cells for the production of antibodies and learned that in addition to antigen and receptor or the membrane-bound antibody binding, there's also a role in CR2, which acts as a co-receptor for B cells that binds to the C3d compounds that are a byproduct of complement. So I like how that last point kind of bookends the interrelationship between antibodies and complement. How complement can act as both an activator for B cells to produce antibodies, and then how antibodies can augment the activation of phagocytosis, through which they can start the complement pathway, and then also by co-binding the CR1 or the MAC receptors with complement with the Fc gamma receptors that can totally help augment the reactions of phagocytes. So that closes out our discussion on how T cells and B cells are activated and their effector functions. And now we're going to get into some of the ways that the body uses the effector functions to fight off cancer, biomaterials, or transplanted tissues, how they're ineffective in the cases of autoimmune functions, and so on. So thanks for listening.